In my last lecture, we have discussed about the mitosis. Now we will discuss about the cell cycle checkpoint. Okay. So before we move forward to understand about the cell cycle checkpoints, first of all, we will understand why the cell is being activated so that it will undergo the process of cell cycle or the process of cell division. So there are many growth factors when they are binding to the receptors, and the growth factors you can also call them the mitogens because they induces the mitosis. So these growth factors, once they are binding to the receptor, they are activating a signaling cascade. Signaling cascade means one molecule is going to be activated, then the another molecule is going to be activated, like this. There is going to be the signaling cascade. For example, if suppose this is your growth factor, it is binding to the receptor, let's say, and suppose this is a G protein coupled receptor. This is the 7-pass membrane proteins, and G protein coupled receptor, this is what the G protein coupled receptor. So G protein coupled receptor, this is a 7-pass transmembrane protein. It is of 3 types. One is the GQ, one is the GS, and one is the GI. These are the stimulatory proteins, receptors, and this is the inhibitory receptor. So once this, and this is the growth factor. So once this growth factor is binding to this growth factor coupled receptor, and suppose this is the GQ protein, G protein coupled receptor, then that will activate the phospholipase C on the cell membrane. Now this phospholipase C will convert the PIP2 to IP3 and diacyl glycerol. PIP2 is phosphatidyl inositol diphosphate and IP3 is inositol triphosphate. These are the lipid molecules. Usually you know the cell membrane is composed of lipid molecules. So what is a PLC is going to do? It will convert the PIP2, because this is an enzyme. This will convert the PIP2 to IP3 and the diacyl glycerol. Now what will this diacyl glycerol do? That will activate protein kinase C, and this is the protein kinase because it's a kinase, so it will phosphorylate the other proteins. And like this, these phosphorylated proteins will again activate certain transcription factors, which we will discuss later on. Now diacyl glycerol is activating the phosphokinase C, and this phosphokinase C, once it is activated, it will phosphorylate the other proteins, and now these proteins, once they are activated, they will activate other proteins, and this is known as the signaling cascade. Now what IP3 will do? IP3 will enhance the release of calcium from the calcium storage center. For example, you can say the rough endoplasmic reticulum. Now this calcium, excess of this calcium will bind to calmodulin receptor, which is a very specific receptor for the calcium, and once they are forming the complex calcium and calmodulin, they are again activating the many other transcription factors or genes. Okay. So this is regarding the GQ stimulatory G protein coupled receptor. And there is a GI, which is an inhibitory G protein coupled receptor. The function of GI is to maintain or regulate the level of calcium. Usually inside the cell, the calcium is in the highly regulated conditions. Otherwise, what will happen? If the GI is not present, calcium level will increase inside the cell. It will activate, it will bind to its receptor and will activate genes, which is not necessary. So this calcium labels, the calcium label inside the cells is under the highly regulated conditions. And how it is going to be regulated? It is going to be regulated by the GI. So what happened now? The growth factor is binding to the G protein coupled receptor, for example GQ type of G protein coupled receptor, that will activate the phospholipase C, and phospholipase C, because this is an enzyme, this will convert PIP2 to IP3 and diacylglycerol. Diacylglycerol will activate the protein kinase C, and then protein kinase C will phosphorylate the other proteins. Wherever the word kinase is used, it means the proteins are going to be phosphorylated. Then the IP3 is the inositol triphosphate. It will enhance the release of calcium from the calcium storage center, and this calcium will bind to the calmodulin, and then again they will activate a certain genes which are required for the process of cell cycle or the process of cell division. This is one way. Now the another way is what? Another way is this: if suppose there are some certain cytokines or certain mitogens, you can say mitogens are growth factors, and once they are binding to their receptor, let's say this is not antibody, let's say this is a receptor. So these receptors, once they are binding, there is another protein JAK. These receptors are going to be phosphorylated, and once they are phosphorylated, there is another protein which is a Janus kinase. That will bind here. Let's say you can take the another color. So you can take, this is your Janus kinase. Now the Janus kinase is binding. Now the Janus kinase is binding to this activated receptor. Now what happened? There is another transcription factor which is a state proteins, STAT. Let's make it like this. Now they are becoming activated. So once the cytokines or mitogens are binding to the receptor, they are being phosphorylated, and to this phosphorylated proteins, this is what is JAK. This is what is STAT. So you can say, so once the receptor is binding to the mitogens, this JAK is being binding to this phosphorylated receptor, and once the JAK is binding to this phosphorylated receptor, STAT is binding to this JAK proteins and forming the dimers, and then again they will activate the multiple genes. This is like the process of signaling cascade. It happened in seconds. This is the another pathway. Let me just clear something here. This is your JAK. Okay, and this is the STAT proteins. Okay, so maybe we should remove this portion also. Okay, so this is the STAT proteins. Okay, so this is the another pathway. Now we will come to the other pathways. Third one, again there were, I already told you, there is another G protein coupled receptor, that is the GS. Once the growth factors are binding to GS proteins, GS is the G protein coupled receptor, then GS, these are the stimulatory protein, they will stimulate the protein or enzyme which is the adenylate cyclase. This adenylate cyclase, what it will do, it will convert ATP to the cyclic AMP, and this cyclic AMP will activate protein kinase A. And again, because there is a word called kinase, is protein kinase A, because there is a word which I am discussing about kinase. So kinase will add the phosphate group to other proteins, and again they will activate certain other proteins which are required for the cell division. So P means phosphorylate, they are phosphorylating the other proteins. Okay, so once the growth factors are binding to G stimulatory type of G protein coupled receptor, they are activating the adenylate cyclase. Adenylate cyclase will convert ATP to cyclic AMP, and this cyclic AMP will activate protein kinase A, and protein kinase A will phosphorylate the other proteins which are required for the cell division cycle. So this is another pathway, you can say. And there are other pathways also, for example the tyrosine kinases. Very important receptors. Most of the research are going on these receptors. Basically what happened, so once the growth factors are binding to these receptor, which is the TKR, tyrosine kinase receptor, they will activate certain other protein GRB2, then they will activate the RES protein. RES will activate the other protein which is the REF protein, and these proteins will activate the very important pathway which is the MAP kinase pathway. Again the word kinase is coming, so it will again phosphorylate the other proteins. And once these proteins are activated, they will activate other proteins, and there will be the phenomenon of signaling cascade until and unless the cell is not undergoing the cell division, because that's what these growth factors want. These growth factors want the cell to divide. So these tyrosine kinase receptor never work alone, they work in the dimeric form. Until and unless there is no dimeric form, okay, they will not work. So once the growth factors are binding to these receptors, they need to form the dimers to be activated, and once they are activated, they will activate the other proteins, specifically the MAP kinase protein, which will activate the other proteins, and that will lead to the cell cycle. So that is how the cell cycle is going to undergo. Now you see these all kind of growth factors. Once the cell is getting a signal that it should divide, now the cell will undergo the cell cycle. And how it will undergo the cell cycle? Because these growth factors are activating transcriptional factors. certain enzymes, certain genes. Okay. So for example, there is a gene called c-MYC, COS, JUN. JUN is of many types, JUN C, JUN D. There are many isoforms of JUN proteins, and once these proteins are activated, they activate certain transcriptional factor. Transcriptional factors always bind to the DNA, so they will bind to the particular sequence of DNA, and they will activate this sequence, and this DNA, this particular sequence, code for the protein which is the cyclin protein. Okay, and cyclins are of many types. For example, they can be of D type, E type, A type, and B type, and they control the transition of cell to G1 to S phase. Then this is the mitotic cyclin. This is the S to G2 phase. So there are many kind of cyclins which control the cycle of cell cycle, basically different phases of the cell cycle. So that we'll discuss later on. Now the cyclins are there, and there is another genes that code for the cyclin dependent kinases. Okay, which is known as the CDK. Cyclin cannot work alone. CDK cannot work alone. They need to come close to each other to function. So what happened here now? So you now have a, let's say this is a cyclin, and let's say this is your cyclin dependent kinases, which is the CDK. Okay. Now this cyclin dependent kinases is going to be phosphorylated at certain site. One is the inhibitory site, one is the activating phosphate. So CDK activating kinase, what they will do? Now they will phosphorylate this protein, let's say at the activated site. Let's say this is the activated site. This is the activated site. Okay. Now there is another protein which is the V1 protein. This V1, what it will do? It will phosphorylate this CDK at the inhibitory site. So once the phosphorylation occurs at the inhibitory site, this whole complex is not activated. So now this inhibitory phosphate needs to be removed, and how it is going to be done? This is going to be done by the CDC 25, which is the phosphatase. Okay? And this is basically activated by some other proteins which are the polokinases. Okay. So here you can see this is the cyclin, this is the cyclin dependent kinases. Now this cyclin dependent kinases can be phosphorylated at two sites. If it is phosphorylated at the inhibitory site, it cannot function, but if it is activated at the activated site, it can function. So what CAK and V1 are doing, CAK is CDK activating kinases. So this will add the phosphate group to the activated site, but at the same time V1 will also add the phosphate to the inhibitory site. Now this complex cannot function. So another enzymes will start functioning at this position, CDC25, which is a phosphatase. So this phosphatase will remove this phosphate group, and now this is going to be only phosphorylated at the let's say activated site. Now this complex is activated and the cell can progress towards the cell cycle phases. So these two proteins are very important, V1 and CDC25, and you should remember that. So once this is phosphorylated, what it will function then? Once this is phosphorylated, it will phosphorylate, because this is what the CDK, cyclin dependent kinases. Again, they are kinases, so they will add phosphate group to the other proteins. Now there is another protein which is the retinoblastoma protein, and that is coupled with the, let's say, E2F transcription factor, and this is the retinoblastoma protein. So this activated CDK that will phosphorylate the retinoblastoma protein, and once this is phosphorylated, E2F is going to be released. E2F is free now, and this is going to be degraded, phosphorylated retinoblastoma protein. Now this E2F is free. E2F is the transcription factor. It can bind to many genes. It can bind to many genes which are required for the progression of the cell cycle. Okay, so that will activate certain genes which may require for the synthesis of DNA, for the replication of DNA. That may also produce genes which are required for the cell to enter in the mitotic phase. That depends. Okay? There are multiple types of the E2F factors. Okay, so this is like that. So you understand here that this is cyclin, this is a CDK. It is going to be phosphorylated by the CDK activating kinases at the activated site. V1 will phosphorylate at the inhibitory site. This inhibitory phosphate is going to be removed by the CDC25. CDC25 is the phosphatase, and now this complex is activated. This will activate the retinoblastoma protein, phosphorylate the retinoblastoma protein, will release the E2F transcription factors, and these transcription factors can bind to the certain genes which are required for the progression of cell cycle. Okay. In this process, cyclin D and the cyclin E plays a major role in the activation of E2F transcription factors. Okay, that you need to remember, cyclin D and E. So now the cell, suppose the E2F is activating certain genes which are required for the DNA replication, and now your DNA is starting replicated. Okay, okay. Now this is a semi-conservative type of replication, but suppose if there is any error, that cell should not undergo the cell cycle process. It should not undergo, it should not enter in the G2 phase or in the M phase. Now what happened? There are proteins which are known as the ATM proteins. ATM proteins are generally work as a proofreader proteins. Okay, so these ATM proteins will read this DNA, and if they found some errors, they will activate p53 protein. Okay, so regarding the p53 protein, this is the p53 protein, which is a tumor suppressor protein. It binds to another protein, which is the MDM2 proteins. Okay, so this is basically the MDM2 proteins. So once they are binding, this protein is binding to the MDM2 proteins, it is not activated. It is in the inactive stage. Okay, once the ATM is finding some error, it will activate the p53. It will phosphorylate this p53 proteins. Let's take the red one. It will phosphorylate the p53 protein. And once this is going to be phosphorylated, this MDM2 can no longer bind to the p53 protein. So now, once the ATM is phosphorylating this p53 protein, MDM2 cannot bind, and now the p53 is activated. Now the p53 protein will activate certain DNA repair enzymes. Once it is activated, it will activate certain DNA repair enzymes. So DNA repair enzymes, they need to repair the DNA. So they will repair the DNA. Okay. And if suppose the cell wants some more time to repair the DNA, then what they will do, this p53 protein, this will activate the other proteins like p21, p27, p17, like this, p18. Okay, if the DNA repair needs more time. So there are two conditions. p53 can activate the DNA repair enzymes, and if the DNA is repaired at the particular time and cell doesn't need time, then it's fine, DNA is repaired, okay, then you can proceed, the cell can proceed for the cell cycle. But suppose if the cell require more time to repair the DNA, then these p53 proteins will activate these other proteins. And what these proteins will do, this is the cyclin protein, right? And this is, suppose, let's say is the CDK. This is, let's say, the CDK, and this is the 6 cyclin. These proteins will bind here. Make it more clear. So what they will do, if suppose this is a this is your cyclin protein, and this is your CDK proteins. And you can take, this is the p27, p17, p18, p21 protein. They will bind to these sites, to the activated site of this CDK proteins, and then this complex becomes inactive. And once this is inactive, it cannot phosphorylate the retinoblastoma protein. Retinoblastoma protein will bind to the E2F factor now, and E2F is not free. It's not free. E2F is not free now. Why it is not free? Because this complex is inactivated. CDK is inactivated. It cannot phosphorylate the retinoblastoma protein, and in that case, what will happen? E2F is not free. E2F cannot bind to the certain genes which are required for the cell cycle. So in that case, these proteins are providing some more time to the cell so that the cell can undergo for the repairing of process. Okay. Now suppose if uh, the repair is not completed, or if the repair is not done in a perfect way, and there is still a mutation, then what the cell should do? Cell should die. So these p53 proteins will activate the wax protein, okay, or BCL-XL protein. These proteins are the pro-apoptotic proteins, and these proteins functions in the intrinsic pathway of the programmed cell death, where the mitochondria, from the mitochondria, these proteins are activated, then the cytochrome C is going to be released, and that cytochrome C will activate the caspase. This is the intrinsic pathway. So if suppose the cell is unable to repair the DNA somehow for any reason, then the p53 will activate these proteins, which are the pro-apoptotic proteins, and will guide these proteins, so that these proteins will guide the cell that cell should die now. It should not proceed for the another division of the cell cycle. So that's how they are working on to repair the DNA during the S phase. So so many proteins are required in the cell division of a single cell. Now one more thing is, DNA replication occurs just once in cell cycle. Okay, it should not be repeated. One DNA should not replicate 2 times or 3 times. Okay, otherwise your cell will have not 2n copies of DNA, it may have 4n or 6n copies of DNA, which we do not want. So DNA is going to be replicated just once. And how? So there is a origin replication complex, and this origin complex, origin, which is also known as the ORC, required. This is the origin center for the DNA replication. At this point only the DNA starts its replication. Now this complex will bind to CDC6 proteins, and it will also bind to MCM proteins. These two proteins it is going to bind. Okay. Now all together this complex is known as the replication complex. Okay, now once this is formed, after that what happened, once the cell is moving forward, this CDC6 is going to be degraded. By the proteases or by the proteasomes. So once this CDC6 is going to be degraded, and if there are any excess of the MCM proteins could be of many types, 2, 3, 4, like that. If there are excess of the MCM protein, that will also going to be removed. So the cell will keep the amount of MCM proteins under regulation. It will also remove once the pre-replication origin complex formed. After that, the cell gets signal that the DNA should replicate now. But at this moment, once the cell is getting its signal, CDC6, the cell will remove the CDC6 from this origin replication, the pre-replication origin complex, and it is going to be degraded, and if there are excess of the MCF proteins, that will also be removed. Now this pre-replication complex will only contain here, once the CDC6 is removed, it is only containing ORC and MCM. And now the S cyclin will activate, which is the cyclin E and cyclin A. These are the S cyclins. They will activate and DNA is going to be replicated only once. So for the process of DNA replication, it is essential for the formation of the pre-replication origin complex. So once the cell is undergoing the process of replication and the CDC6 is removed, it cannot undergo the process of another cell replication, DNA replication. So DNA will only replicate just once, because the cell is removing the CDC6 proteins from the pre-replication origin complex. Okay, once this is removed, this complex is inactivated, and once this is inactivated, the process of DNA replication one more time cannot be happened. So this is why the DNA replication occurs just once. This is the explanation of this, and you can understand how many proteins are working all together in a perfect way. So DNA is replicated just once. Okay. Now coming to the cyclins, cyclin D, cyclin E, cyclin A, and cyclin B. You can say DAB, DEAB, DAB. So D is Sajivan cyclin. It has its preferred partners, which is the CDK4 and CDK6. There are many inhibitors of CDK4 and a CDK6 on which the clinical trials are going on to prevent the cancer, and you can Google and you will find it out. Then there is a CDK cyclin E. This is the S cyclin. Or you can say G1S cyclin, that bind to the CDK2. So certain inhibitors, whatever they are in the market, they are not pan inhibitor. They will not inhibit all these proteins. Some are like very specific. They will inhibit only CDK4 and the CDK6, but not the CDK2. Then cyclin A is the S cyclin. Again, it has the preferred partner of the CDK2, and the cyclin B, which is the mitotic cyclin, and it has its preferred partner of CDK1. So you can remember like that, 4, 2, then this is again 2. Four, 6, 2, and 1. The irreversible order of numbers. Okay, like this. And DEAB you can remember like DAB, DEAB. So you can remember that. Okay. So these are the cyclins which are the cell cycle checkpoints. Now there is another protein which is the APC protein. This is the anaphase promoting complex. And this protein binds to CDC25. This is the checkpoint of metaphase to anaphase. This is CDC25. Let me just see if I can. Okay, so this CDC25, what it will do, there is a protein called securin that is bind to the CSIR protein. So this anaphase promoting complex, once it is activated after binding with the CDC25, it will add the ubiquitin chain to this securin protein, and that will lead to the proteolysis. So this protein is going to be degraded, and now the CSIR is free. And then CSIR, what in the anaphase condition, you know, the chromosomes are like this, but in the in the metaphase condition, the chromosomes in like this, and in the anaphase condition they will separate. And how they will separate? Because of this process, the anaphase promoting complex will add the ubiquitin chains to this securin protein, and thus this securin protein, once the ubiquitination is done with the help of ubiquitin ligase, then the proteasomes will activate and will degrade this protein. Now the CSIR will free, and CSIR will separate these sister chromatids. Okay, now this is like the CDC25. Now we will come to the another thing is like the CDC25 and the APC complex. The another function of this protein is to degrade the M cyclin. Okay, this is very essential. Once the cell is, suppose if you have the M cyclins which are non-degradable, suppose you are doing any mutation in these genes or in these proteins, and they become non-degradable, the cell will permanently arrest in the mitotic phase. Cell will not enter in the G1 phase. Okay, so this is important. So now this CDC25 APC need to degrade the M cyclin so the cell can enter in the another round of the cell cycle, which is the G1 phase. But the thing is, once the cell exits from the mitosis, then this protein, that these M cyclin, they again accumulate, and the concentration of these M cyclins again it is started increasing. Okay, so what happened, there is another protein which is the HEC1 APC. Once they starts to accumulate, these proteins becomes activated, and then these activated proteins will again degrade these M cyclins. So the fundamental rule is this: the M cyclins need to degrade it at the end of mitotic phase, but they should not start accumulating immediately after the exiting of mitosis, because if they are immediately, if the concentration of these M cyclins is increasing immediately, the cell may not enter in the G1 phase. It will directly enter into the S phase. So it is essential that these M cyclins need to be at a very low concentration or very in the inactive stage until the cell is coming to the mitotic phase. Therefore the HEC1 proteins will become activated, and once the M cyclin proteins starts accumulating in the G1 phase, HEC1 proteins will degrade them. So it is like this. This is, suppose this is your CDC25 and APC, which will less in the G1 phase, or after exiting from the mitotic phase, there is an increasing concentration of HEC1 and APC. So HEC1 and APC will degrade the M cyclin, because we don't want M cyclin to be accumulated in the G1 phase. They should only become active at the mitotic phase. That's what we want. When the cell is in the mitotic phase, then they should activate it. And M cyclins are nothing but the cyclin B. So this is regarding the cell cycle checkpoints, and in the next lecture we will discuss about the microbial physiology. If you like my channel and if you have any comments so that I can improve my content, I would be happy to do that. Please subscribe my channel if you like the content. Thank you so much.